Dear students, the topic is about altered gene functions. This is mainly discussing about mutations, the altered gene expression. Okay, and this mutation is firstly introduced by Hugo de Vares in 1901. There is a phenotypic changes which are produced in the one of the primrose plant. And here the mutation is permanent alteration in genetic material. That means the permanent changes that occurs in the DNA sequence. The mutation that affect the germ cells, specifically sperm or the ovum, and are transmitted to progeny and may give rise to inherited diseases. Here, the mutations that affect somatic cells are not transmitted to progeny but are important in genesis of cancer and congenital abnormalities. About the introduction part, the genetic alteration mainly include the chromosomal abnormalities and gene mutation. Here, as I said earlier, the gene mutations are permanent changes in the DNA gene sequence, and that is arise during the normal DNA replication or in response to the certain environmental factors. So there are many classes of gene mutations, and certain mutations can cause diseases. So about the altered gene function, that is mainly refers to the changes in the normal expression, regulation, or activity of a gene that results in a modified cellular phenotype. So, in case of pharmacological context or in biomedical context, this concept is central to disease pathogenesis, drug response variability, and therapeutic targeting. And there are certain mechanisms are there for the gene altered gene function, and in that first one is the genetic mutation. So the genetic mutations are permanent changes in the DNA sequence that affecting the gene structure or expression. So in case of biology, mutation is an alteration in the nucleotide sequence of the genome of an organism, virus or extra chromosomal DNA. So here this mutations results from errors during DNA replication, mitosis, meiosis or other types of damages to DNA, which then may undergo error-prone repair or cause an error during other forms of repair or else may cause an error during replication. That means in case of translation process. So that mutation may also result from insertion or deletion of segments of DNA due to mobile genetic elements. So we have different types of mutations that are point mutations, then insertions, deletions, or insertion or deletions, or copy number variations. Okay. So in this slide, we are discussing about different types of mutations. The mutations are classified into point mutation, that is base substitution mutation, which include silent mutation, missense mutation, nonsense mutation. And other one is the frame shift mutation, then trinucleotide repeat mutation. And about this point mutation, and that first is the missense. Here, simply we can say that there is an altered amino acid so that the modified protein function occurs. Then about nonsense mutation, here the premature stop codon is produced and truncated protein produced. In case of silent mutation, there is no amino acid changes but may still affect the splicing. And next is insertions or deletions. Here, they can cause frame shift mutations, drastically altered protein occurs. Then next is the copy number variations in that gene amplification. Either it can increase the expression or deletion. Deletion means there will be a loss of function occurs. This slide is explain about gene mutation. In that about the point mutation. Here this point mutation means substitution of a single base by another is known as point mutation. And here this transition. Transition means a purine base may be replaced by another purine base or pyrimidine base may be replaced by another pyrimidine base. And transversion. Here a purine base may be replaced by a pyrimidine base or vice versa. So point mutation can affect only one codon, and these point mutations are three types, as I said. or that is silent mutation, missense mutation, or nonsense mutation. And in that, this silent mutation means there is no change in amino acid that is due to codon redundancy. And in case of missense mutation, one amino acid is replaced by another. It can alter the protein function. And nonsense mutation means codon changed to stop codon. That means a truncated protein are produced. Then about frame shift mutation. Here, addition or removal of nucleotides occurs. In that, if not multiplies of threes, that is a frame shift mutation, that means alters the reading frame, that is drastically different protein sequence occurs. And based on size and chromosomal involvement, small scale mutation occurs, then large scale mutation occurs. And in case of this small scale mutation, point mutations and small indels, it can affect one or few nucleotides. In case of large scale mutations, deletions means a loss of chromosomal segment occurs, duplication means a repeated DNA segment, then inversion means segment is reversed, translocation means what will happen, segment move to another chromosome. So these often affect multiple genes and are clinically relevant or clinically significant. And there are based on functional consequences. They are loss of function mutations, then gain of function mutations, then dominant negative mutations, lethal mutations. So about the substitution mutation as a single nucleotide alteration in which one base pair in DNA is replaced by another. So despite involving only one nucleotide, its impact ranges from negligible to profound and mainly depending upon its position, codon context, and effect on protein structure or its function. So based on the molecular basis and its mechanism, substitution mutations are basically arise due to errors during DNA replication. That is misincorporation by DNA polymerase. Then spontaneous chemical changes that occurs, that is mainly due to deamination. That means cytosine to uracil or depurination. Then mutagen exposure can affect. That means certain chemical agents like alkylating agents or base analogs exposure or radiation can cause mutation. And here the substitution mutation is that exchange one another for one base pair for another. That is, it changes in single chemical nature, such as switching to an A to G, that is adenine to guanine. And here the change in codon to one that encodes a different amino acid, and that can cause small changes in the proteins which are produced. For example, sickle cell anemia is caused by substitution in the beta hemoglobin gene and which alters a single amino acid in the protein produced. And here this change a codon to one that encodes the same amino acid and causes no change in the protein produced. That we can call it a silent mutation. So this is an example for substitution. What will happen here? C, T, G, G, A, and G. This is the sequence of the one codon. And another here, what will happen? C, T, G, G, and G. Instead of adenine, it is replaced by guanine. This like this, the substitution can occur. That is, errors during the DNA replication, or there is a spontaneous chemical changes can occur, or sometimes the mutagen exposure can leads to the changes. This slide is also remind you what is point mutation. So this is also known as base pair substitution. Change a single nucleotide base pair. There are three types of point mutations are there. That is, first is silent mutation, missense mutation, and nonsense mutation. We'll discuss detail about the silent mutation or synonymous uh, mutation. So this silent mutation is a change in one DNA base that does not change the amino acid in the protein. So the protein remains the same. So why does it happen? Because of the genetic code, one amino acid can be coded by multiple codons. A change in the codon may still give the same amino acid. For example, codon is guanine, then adenine and adenine. Amino acid is the glutamic acid. And next is guanine, adenine, and guanine. Amino acid will be the glutamic acid. If it is guanine, adenine, adenine to guanine, adenine, guanine, the amino acid is still glutamic acid. So this is what is called a silent mutation. So here we have to remember usually this occurs at the third position of the codon, that means known as vowel position, and there is no change in the protein sequence, hence it is called it as silent. So it is always completely silent. That's a question. The answer is not always. Sometimes it can and change how fast a protein is made, or sometimes it can affect the mRNA stability. So sometimes it slightly influence the protein folding. But in almost in all the contexts, it is named as silent mutation means there is no change in the amino acid. So, in one line, we can say that a silent mutation is a point mutation in which a codon change does not alter the amino acid due to the degeneracy of the genetic code. Mutations, that is silent mutation. Let us take double sequence of DNA and the mRNA, which calls for the codons and which gives rise to the amino acids. And here you can see that is there is no mutation occurs. And next is the transition mutation, that is purine changes. For example, first codon is not affected, but second codon is usually there is a hypothesis called bubble hypothesis, and here there is a third codon is changed. A is replaced with G and purine to pyrimidine. And so the complementary nucleotide is changed in the case. And the RNA is code is changed, but the amino acid is not changed. So this hypothesis is known as Vogel hypothesis, and where there is a degeneration of the genetics occurs. For every amino acids, according to the amino acids are available. And so that here the third codon is not affected, so that there is no change in the final protein sequences. And next is a transverse mutation. Here this purine to pyrimidine change occurs. For the first codon, next codon is first two is not affected, and third one is changed, and that will be changed to pyrimidine. Instead of purine, it's changed to pyrimidine. What happens, the complementary nucleotide is get changed and the codon is totally get changed, but the amino acid will be the same, and the third codon is not affected, and the final protein sequence is not changed. These type of mutations are called silent mutation. So this is about nonsense mutation. That is as a type of point mutation in which a codon that originally codes for an amino acid is converted into a stop codon. That means that leads to a premature termination of protein synthesis. And here about normal DNA codon that is basically code for the amino acid. And in case of nonsense mutation, here this codon becomes a termination codon. And there are basically three stop codons are there. First is UAA, then UAG, then UGA. These are the three major termination codons are there. And its mechanism means that basically this is occurs due to single base substitution. For example, normal codon for the methionine is in case of DNA, first codon will be that is a TAC, then mRNA is produced, and then AUG. And in case of mutated one, here DNA codes for ATG and converted to mRNA and stop codon is produced. So what will happen? The translation as it stops prematurely and truncated protein gets produced. And here, in coming to the molecular consequences of nonsense mutation, here it produces a shortened or a truncated polypeptide. And usually, what will happen? It is non-functional in its nature, and that can lead to loss of protein activity and rapid degradation through the nonsense-mediated mRNA occurs. And about its pharmacological relevance, here this what nonsense mutations are basically implicated in several genetic disorders. And first is Duchenne muscular dystrophy. What will happen here? The premature stop in the dystrophin gene occurs and non-functional protein produces. Then next is you are all familiar with the cystic fibrosis. Here some cases involve nonsense mutations in CFTR gene. And next is in case of beta thalassemia, an early stop codon gets produced and defective globin synthesis occurs. About its pharmacological importance, what happens? It's a targeted for the precision medicine. For that, for example, atallurine that can promote ribosomal read through a premature stop codon, so that it allows the production of partially functional protein. And you have to remember that this nonsense mutation is a substitution mutation. The effect is premature stop codon formation occurs in this case, and major outcome is a shortened or truncated, usually non-functional proteins get produced, and basically this is associated with severe genetic diseases, and these are the main points which are regarding nonsense mutation. This is about point mutation, that is missense mutation, that is acceptable is the in that codon containing the changed base codes for different amino acids. The sequence of amino acids in the protein changes. Acceptable missense mutation, here the substitution of amino acid has minimal or there is no effect. For example, uh, hemoglobin A has valine at the position 67 of beta chain, but hemoglobin Milwaukee here glutamate is at the position of 67 of beta chain in place of valine. In case of hemoglobin Bristol, aspartic acid is at the position 67 of beta chain in place of valine. Then in case of hemoglobin Sydney, alanine at the position 67 of beta chain in place of valine. But there is no change in their action. So this frame shift mutation shifts the reading frame of mRNA due to insertion or deletion, leading to a completely altered and usually non-functional protein. So this mutation occurs when the addition or loss of DNA bases changes a gene's reading frame. And this reading frame consists of a group of three bases that each code for one amino acid. So a frame shift mutation shifts the grouping of these bases and changes the code for amino acid. This resulting protein is usually non-functional or truncated proteins are produced, and that is mainly due to insertions, deletions, and duplications can also also be a frame shift mutation. This is about the frame shift mutation. That is a genetic alteration which is caused by the insertion or the deletion. That is known as indel. That is insertion and deletion. In short, we can say that indel of the nucleotides that are not in the multiples of 3, resulting in a shift in the reading frame during the translation. And about its basic concept means generally the genetic code is read in triplets. That means that we can call it as codons. If the nucleotides are added or removed, that is instead of three codons, the entire downstream codon sequence get changed. And for example, in case of normal sequence for the DNA, ATG, then AAA, then GGC, then TTT. And in case of for that mRNA, that is AUG, AAA, GGC, then UUU. So that is named as met lysine, glycine, and pH. Okay, and after the deletion of one base, what will happen? This instead of this met lysine ALA, a wrong sequence get continuous. Instead of double last codon UUU, you will get only two UU. What will happen? There will be a shift occurs. So completely different amino acids from that point onwards occurs. Shifting will be there. So the molecular consequences means alters downstream codons, and often it can introduce a premature stop codon development or the protection, and so that it can produce a severely truncated protein or an abnormal elongated protein, usually results in the loss of function. And mutation is basic character in this case of frameshift mutation. Here insertion or deletion occurs, and not in the multiples of 3, that can be 2 or into 1. And major effect is severity will be high, more disruptive than the missense mutation. And here important example for the pharmacological relevance is you are all aware about Crohn's disease. Some cases involve this kind of frame shift mutation in NOD2 gene, so that it can leads to a Crohn's disease. And sometimes BRCA1 associated breast cancer. Here sometimes frame shift mutation occurs and results in a truncated tumor suppressor protein. And important pharmacological significance of these kind of frame shift mutation is important in case of genetic diagnostics, cancer genomics, then drug targeted identification. And emerging case is the gene editing. That is, you're all aware about CRISPR technology and gene replacement therapy. So in case of frame shift mutation, the major cause is a deletion or insertion of the three base pairs, and a net effect is it can alter the entire reading of the frame, and there will be a frame shifting will occur, and completely abnormal protein is produced. And in the case of severity, it's a very high severe cases are there compared to nonsense mutation. And frame shift mutation shift the reading frame of mRNA due to the insertion or deletion. It can lead to a completely altered and usually non-functional protein gets produced. So about this mutation. Insertion and deletion mutations are structural changes in the DNA that are involving the addition or removal of nucleotides, collectively termed as indels, and their biological impact depends critically on whether they preserve or disrupt the reading frame. And coming to the definition for this insertion, addition of one or more nucleotides into a DNA sequence is known as insertion. And normally, for example, normally DNA ATG, AAGGC, and after insertion, one base is also added. In that, what will happen? A DNA ATG, CAA, GGC. So that is, it can alter the codon grouping, so that may shift reading frame. And next is about the deletion mutation. Here, the name itself, removing of one or more nucleotides from the DNA sequence. For example, normally DNA will be ATG, AAA, GGC. After deletion of the one base, what will happen? DNA, that is ATG, AAG, GC. So it can also cause the misreading of downstream codons. And that is the functional classification of frameshift index, that is the most critical one, that occurs when insertion or deletion of multiples of three codons occurs. So that are results in the reading of frameshift, alter downstream of amino acid sequences, often premature stop codon preparation. So clinical severity will be high in this case. Then in frame index, it occurs when insertion or deletion of multiples of three nucleotides occurs. So that can also leads to the addition of removal of the specific amino acids, reading frame preserved. Then functional impact means it will be variable from mild to severe depending upon the protein domain which is get affected. And insertions of mutations in which that means simply you can say that there will be an extra pairs are get inserted into the nucleus in the DNA. So, and this insertion changes the number of DNA bases in a gene by adding a piece of DNA. And during this deletion means the name itself, we can remove a piece of DNA. Insertions or deletions may be small. That may be one or few base pairs within the gene. A large in the sense the entire gene or the several genes or the larger sections of the chromosome is get deleted, or in any of these cases a protein made by the gene or may not function properly. And deletions are mutations which a section of DNA gets lost or gets deleted. In this picture you can see here insertion means CTGGAG is there, but in into that one pair of that means three codon will be get inserted, that is CTGG. After that TGGAG get inserted, that is known as insertion mutation. About the deletion mutation, a deletion changes the number of DNA bases by removing a piece of DNA, and the small deletion may remove one or few pairs within the gene. And what will happen during the larger deletion can remove one entire gene or the several neighboring genes, and the deleted DNA may alter the function of the resulting protein. There will be a starting sequence will be there in between the deletion of the cordon will be there. And majorly this example for the frame shift, frame deletion, that is in case of cystic fibrosis, there will be a in frame deletion occurs, that is a loss of phenyl aniline occurs, there will be a defective CFTR folding occurs. And also in the case of Huntington diseases, there will be expansion, that is insertion occurs of CAG repeats, and toxic gain of functional protein occurs that can lead to Huntington disease. And in case of pharmacological and therapeutic significance of these insertions or deletions, and mainly molecular diagnostic purpose, we are using PCR sequencing, uh, then precision medicine, and therapeutic strategies that are include gene editing, that is CRISPR Cas9 for the correction of intelts, and protein modulators in case of CFTR characters in the cystic fibrosis, and antisense oligonucleotides for the exon skipping, that is dystrophin for the restoration. uh, and majorly the insertion means you have to remember that there is an addition of nucleotides occurs. Deletion means removal of the nucleotides, and each depends on the reading frame preservation. And frame shift index, that is the most severe cases. And this in frame index can variable impact will be there from higher to severe to mild, and stronger association will be there for the with the genetic disorders and pharmacogenomics. So, insertion and deletion mutations definitely can alter the DNA by adding or removing the nucleotides. And when not in the multiple of three, if they can cause frame shift mutation leading to the major protein dysfunction. This is about the base. This is about base substitution mutation, in which one base pair is replaced by another. So this base substitution mutation is a type of point mutation in which one nucleotide base is replaced by another in the DNA sequence without altering the overall length of the gene. And about the basic concept here, what will happen? One base pair is substituted, and there are two types of base substitutions are there: transition mutation and transverse mutation. And in case of one base pair substitution means purine is get substituted by the another purine. Example, adenine is substituted with a guanine. And here pyrimidine is substituted with the another pyrimidine, and here does not shift the reading frame. Okay, and there are different types of base substitution. Silent submutation will be the, that means codon change, but the same amino acid will be the due to degeneracy of the genetic code. For example, here both codons, that is GAA and GAAG. So both code for the glutamic acid, so that if there is any changes in the codon, there will not be any change in the protein function. So this is about transition mutation. It's a type of point mutation where one purine base is replaced by another purine, or one pyrimidine is replaced by another pyrimidine. For example, purine is adenine guanine, and pyridine is cytosine. Instead of cytosine, thymine is replaced. So the transitions are A to G or C to T. Okay, and transitions are more common than the transversion, and that is purine to pyrimidine changes are transversions, and they often cause silent mutations. That means there is no amino acid changes occurs, and because of this redundancy in the genetic code. And but they can also lead to missense mutations, that is different amino acids, or nonsense mutations means at the time stop codon gets produced, and that are all depending upon the codon which is affected. So about this transition mutation usually occur due to tautometric shift, that is temporary structural changes in the bases, or the deamination. Deamination in the sense, for example, for the cytosine, deamination occurs and it forms uracil, and it is read as thymine. And replication errors that is not corrected by proofreading. Okay, then about its clinical relevance are there. That is genetic disorders like cancer mutations are there. These are all involved transitions. Okay, for example, beta thalassemia can result from the transition mutations in the beta globin gene, and in pharmacogenomics, transition mutations may alter the drug metabolism gene. That means, example, CYP enzyme get altered, and that alteration may get affecting the therapeutic responses. Okay, and transitions stay in the same family, that is purine to purine, pyrimidine to pyrimidine, like that. Okay, so here this there will be a transition means is the replacement of a base by another base of the same chemical. Here here we are discussing about transversion mutation. So the transversion mutation is a subtype of base substitution mutation in which purine is replaced by a pyrimidine or vice versa, leading to a structurally more disruptive change in DNA. And DNA base is basically classified into purines and pyrimidines. So it consists of adenine, guanine in case of purine, and pyrimidine will be cytosine and thymine. And the transversion means adenine is replaced by thymine, adenine is replaced by cytosine, guanine is replaced by thymine, guanine is replaced by cytosine. So this contrasts with the transition mutations where the substitution occurs within the same class. That means adenosine to guanine, cytosine to thymine. So here transversions are important because it causes greater distortion of the DNA structure compared to transitions. So it can alter the codon meaning and can produce missense or nonsense mutations, but less frequent but often functionally significant. For example, the normal codon for the glutamic acid will be GAA. The same time the transversion mutation is happen, that is instead of GAA, GTA will be there. That means adenine is replaced by uh, thymine substitution. So that can cause for valid. So it can leads to amino acid substitution. That means missense mutation can occur. Then at the molecular level, what will happen? Missense mutation is the most common outcome of this transversion mutation. And here this nonsense mutation causes stop codon if it is formed, and it can also affect the protein folding, enzyme active site formation, receptor ligand interaction, etc. Then coming to this clinical relevance of this transversion mutation, example as uh, it can implicated in several kind of disorders like sickle cell anemia, that is adenines transversion takes place to thymine and in the beta globin gene, and in case of lung cancer, that is guanine is replaced by thymine. The transversions are commonly associated with tobacco carcinogens. Then its pharmacological significance are the basically it is most important in case of cancer genomics, that is mutation is a signature and drug resistance mechanisms, then pharmacogenetic variability. And here this some examples are there, that is mutations in the drug targets, and it can alter the drug binding affinity. And if we are comparing instead of this transversion and transition, the type is in case of transversion, that is purine to pyrimidine occurs, and this frequency is mostly less common. Structural impact will be higher, and functional effect is often severe. It will be there. So here, subtype of this base substitution is the transverse mutation, and which involves different base classes, and it's less frequent but more destructive in cases. And it also often leads to missense and nonsense mutation. So about the deletion on a chromosome. Here, chromosomal deletion is a structural chromosomal abnormality in which a segment of chromosome is lost, resulting in the absence of multiple genes with that region. Unlike point mutation, this affects large DNA segments and often has profound phenotypic consequences. And loss of chromosomal fragment can involve few base pairs. That means microdeletion, and large segments means visible under karyotyping. Leads to haploinsufficiency, that is single copy of gene is not sufficient for the normal function. And there are different type of chromosomal deletions are there. That is terminal deletion is the in that loss of the end, that is telomeric region of a chromosome get lost. For example, deletion of a chromosome tip, and interstitial deletion, that is intercalary deletion, loss of an internal segment and remaining end rejoin, and that can lead to a shortened chromosome. Then about it's all about the mechanism of deletion of chromosome. Basically, this is caused by DNA strand breaks and faulty recombination during the meiosis, and that is also due to exposure of different mutagens like radiation or chemicals. And major consequences are there will be a loss of multiple contagious genes and can result in the gene dosage imbalance, disrupted regulatory networks occurs, and often leads to developmental abnormalities, severe clinical phenotype. And major clinical relevance will be this majorly many of the disease conditions are formed, and once this Williams syndrome, that is a deletion and affect the elastin gene, and cardiovascular abnormalities can occur or cognitive profile may get affected. And Diegeorge syndrome, that is mainly due to microdeletion on chromosome 22. Majorly cardiac defects can occur or immune deficiency can occur. Then about its clinical significance, here genetic diagnosis microarray technique can be used for this purposes. Then personalized medicine can be prepared. The major implications are altered drug metabolism. If the genes are encoding enzymes, something is get deleted, it can alter the drug metabolism. Complex management due to multi-system involvement are there. And here the chromosomal deletion, and if you are comparing with chromosomal deletion and gene deletion, in case of chromosomal deletion, the larger segment will be get deleted, and major effect is multiple gene get lost, and usually the severity will be severe, and its detection is mainly by cytogenetic genetics or by microarray. But in case of gene deletion, that are involved only few nucleotides, and single gene only get affected. The severity will be variable, and this can be found out by sequencing. And you have to remember that the structural chromosomal abnormality will be the um, major impact of chromosomal deletion, and can cause loss of genetic material, and can lead to haploinsufficiency. Often this is associated with syndromic conditions and majorly diagnosed by cytogenetic or molecular tools. And so you have to remember that chromosomal deletion is a loss of segment of DNA from a chromosome, and that can result in missing genes, and significant clinical consequences are due to the gene dosage imbalance. This, uh, so this slide explaining about the difference between inversion, deletion, and duplication. This these are the structural chromosomal abnormalities involving rearrangement or alterations of chromosomal segments with distinct genetic and clinical consequences. About the definition of inversion, that means a segment of chromosome is reversed in orientation without loss or gain. So here the broken chromosome part joins it again, and but it is turned in 180 degree. So this is the picture showing before and after inversion process. And next is deletion. Deletion means there will be a loss of chromosomal segment, and that means missing the genetic material. So here the chromosome fragments break down, and that will be missing from the genetic material. And next is about the duplication. Here the duplication means a repetition of the chromosomal segment. That means one extra genetic material will be there. So that means simply you can say that in the picture itself it's showing that chromosome segments is get doubling. So the duplication occurs when a portion of a chromosome is repeated, and substantial chromosome duplications can create a condition in which a number of genes are present in three copies rather than the two copies normally present. That is partially we can call it as a particular condition, we can call it as a trisomy. And cellular activities are very sensitive to the number of copies of genes, and thus extra copies of genes that can have serious harmful effects. So this is about types of deletion that are include chromosomal deletions are classified based on the location and size of the lost segment. These distinctions are important for understanding pathogenesis, diagnosis, and clinical severity. And first is about the terminal deletion, the deletion that occurs towards the end of the chromosome. So that is known as or the end of the chromosome means that is the end of the telomere. So here the single chromosomal break is happened and the cell fragment is lost, and often that cause a larger deletions and typically severe phenotypic consequences are occurs. And this is the example for Cry-Du-Chat syndrome. That is a deletion of the short arm of the chromosome 5 occurs. Then next is the intercalary deletion or interstitial addition, the deletion that occurs from the interior of the chromosome. So there is a loss of segment from the middle of a chromosome with the rejoining of flanking regions. So here the two breaks are occur and internal segment is get removed and remaining end gets fused together. Maybe the main characteristics are that involve specific gene clusters and variable severity will be there, that is depending upon the genes lost. Example is DiGeorge syndrome, that is the microdeletion affecting the multiple developmental genes. Then next is the microdeletion, that is a very small deletion, a kilobase or to megabase range, not visible under conventional karyotyping. And here this relatively small amount of the deletion occurs, and here the major detections are mainly by rigorous molecular techniques like FISH or the microarray technique or NGS. And major characteristics are there will be a loss of few but critical genes will be get lost and often syndrogenic. Example is Williams syndrome. Then last is the macro deletion or the gross deletion. Here the large chromosomal deletion visible by the light microscopy or karyotyping that are involved, multiple genes are involved, the severity will be usually severe and developmental abnormalities will be happen. And functional classifications are there, that is homozygous deletions are there. Here both copies of the gene gets deleted, usually lethal or very severe. Heterozygous deletions are all occurs, that is by the one copy is get deleted and leads to haploinsufficiency. And so simply we can say that there are different types of deletions are there, that is terminal, interstitial, micro deletion, and macro deletion will be there. So basically these type of deletions are classified based on its location, whether it is terminal, interstitial, and size means whether it is micro or macro, and can cause the loss of genetic material. That means gene dosage imbalance can occur. Often it is associated with various developmental syndromes and required this advanced diagnostic tools for the small deletions. So the chromosomal deletions are classified into, once again I will repeat, terminal, interstitial, microdeletions, and macrodeletions based on their location and size, with clinical severity depending upon the genes lost. There are certain causes are there are certain causes are include, that is losses from the translocation, chromosomal crossovers within the chromosomal inversion, unequal crossing over can occur, breaking without rejoining may occur, for synapses to occur between the chromosomes with a larger intercalary deficiency and a normal complete homologue. The unpaired regions of the normal homologue must loop out of the linear structure into a deletion or a compensation of the loop. So, already discussed what are the effects. Once again, I will repeat that there is a small deletions are less likely to be fatal or it's less severe, but the larger deletions are usually fatal or gets severe. Some size, medium-sized deletions leads to recognizable human disorders. Example, Williams syndrome. In contrast, a deletion that is evenly divisible by 3 is called inframe deletion, and deletions are muscular dystrophy, cystic fibrosis are the common genetic causes in the infant death. So this is about gene amplification. So gene amplification refers to an increase in the number of copies of gene within the genome. Okay, so gene amplification refers to the increase in the number of genes of the same gene rather than to the increase in its rate of transcription. So here So that can lead to the overexpression of the encoded protein. It is a key mechanism in oncogenesis, drug resistance, and cellular adaptation. And normally, the two copies of genes are formed, that is diploid in state. And in case of amplification, multiple genes, that is tens to hundreds, are formed that will result in the increased mRNA transcription, increased protein production occurs. So the gene amplification results from a repeated replication of chromosomal region. In some cases, gene amplification provides a mechanism for increasing gene expression during the development. And gene amplification is also frequently occurs in the cancer cells, and which can result in the elevated expression of the genes that contribute to uncontrolled cell proliferation. And about this gene amplification, it is a major mechanism through which new genetic material is generated during molecular evolution. It can be defined as any duplication of a region of DNA that contains a gene. Gene duplications can arise as a product of several types of errors in the DNA replication or repair machinery, as well as through the fortuitous capture by selfish genetic element. So here the unequal crossing over occurs, and that means during this mechanism of gene amplification, during meiosis, misaligned homologous chromosomes occurs, and that can lead to duplication of gene segments. And in case of DNA replication errors, means what will happen? A re-replication of the specific genomic region occurs. And next is the extra chromosomal amplification, formation of a double minutes, that is DMs, small circular DNA fragments carrying amplified genes. And next is the homogeneously staining regions. So here this amplified DNA integrated into a chromosomes, and which appears as uniform staining regions in the cytogenetic analysis. Important molecular consequences are there will be increased gene dosage will be there, and the overproduction of growth factors, receptors, enzymes. So this maybe leads to uncontrolled cell proliferation or altered cellular signaling. And in case of cancer biology, gene amplification, that is the hallmark of many of the cancers. That is HER2 amplification, that is mainly seen in the breast cancer, that can lead to a aggressive tumor phenotype. And then MYC amplification, that can promote the cell cycle progression. And it also included in the drug resistance amplification of a drug target or a metabolic enzyme genes can reduce a drug efficacy. And major example, this is amplification of dihydrofolate reductase. So this amplification of dihydrofolate reductase can leads to the resistance to methotrexate. And it's also affect the targeted therapy, that is the exploited therapeutically with the trastuzumab, that is a major target for HER2 overexpressing breast cancer cells. And the major detection methods of this is fluorescence in situ hybridization, that is FISH, and then quantitative PCR, qPCR, and then comparative genomic hybridization technique, then next generation sequencing. These are the important techniques that are included in the detection of uh, this defects. And majorly, you have to remember, simply gene amplification means increasing the gene copy number and can leads to protein overexpression, common, which is most common in the cancer and drug resistance. And cytogenetic features are include double minutes and HSRS. Then about the gene amplification and duplication, gene amplification means multiple copies will be produced. Then overexpression will be there. That is relevant for the clinical relevance, means it can involve in the cancer and drug. This diagram simply representing the gene amplification or the DNA before the amplification. And first will be the origin will be very small, and the DNA during the first round interphase replication that will be get enlarged, and second round replication and third round of replication is represented. These are the simple representation of the gene amplification. So the loss of heterozygosity or LOH. So this is a genetic event in which a cell that was originally heterozygous, that is, two different alleles will be there at the locus, loses one allele and resulting in a functional hemizygosity or homozygosity. So here this is common form of allele imbalance by which a heterozygous somatic cell becomes homozygous because one of the two alleles get lost. This form of chromosomes instability is sufficient to provide selective growth advantages and has been recognized as a major cause of tumorigenesis. And this LOH is a cross chromosomal event that results in loss of the entire gene and the surrounding chromosomal region. And this could happen when a part of the DNA is accidentally deleted, perhaps when the cell is undergoing a normal division and replication. The gene might be completely gone, or a part of it might have been moved to another location of the DNA. And all the diploid cell which contain two copies of genome, one from each parent, that is chromosome pair. For the majority of the positions in the genome, the base present is consistent between individuals. However, a small percentage may contain different, and these positions are called single nucleotide polymorphism or SNPs. When the genomic copies derived from each parent have different bases for these SNPs, the region is said to be heterozygous. Most of the chromosomes within the somatic cells of the individuals are paired, allowing for SNP locations to be potentially heterozygous. However, one parental copy of a region can sometimes be lost. Some results in the region having just one copy. And the single copy cannot be heterozygous at SNP locations, and therefore the region shows loss of heterozygosity. Loss of heterozygosity is due to loss of one parental copy in a region is also called hemizygosity in that region. So normal state, one normal, that is wild type allele plus one mutant allele will be there. After loss of heterozygosity, loss of the normal allele, only the mutant allele will be remains. This may be the key mechanism in tumor suppressor gene inactivation. And in case of loss of heterozygosity represents a second hit in many of the cancers. First hit means mutation in one allele only, and during the second hit, loss of heterozygosity means loss of the remaining normal allele. So the classical model will be the Knudsen's two-hit hypothesis will be there. And major mechanism of this loss of heterozygosity mainly by the chromosomal deletion. That means physical loss of chromosomal segment which containing a normal allele. Then mitotic recombination. That means exchange between homologous chromosomes. That means mutant allele is being get duplicated. Then sometimes the gene conversion can occur. Normal allele replicated, sorry, replaced by the mutant sequence. Then a chromosomal non-disjunction can occur. That means here loss of one chromosome followed by duplication of the mutant chromosome gets lost. In case of loss of heterozygosity, in either case protein is encoded by the gene cannot be correctly made. Instead of having the two different versions of the same gene present, that is heterozygosity, one copy of the gene is get lost. And now this could happen when the part of the DNA is accidentally deleted or perhaps when the cell is undergoing normal division or replication. In case of molecular consequences, there is a complete loss of tumor suppressor function occurs that can lead to uncontrolled cell proliferation and genomic instability. About the clinical relevance, here this loss of heterozygosity is strongly associated with the tumor suppressor genes, and here this LOH as loss of heterozygosity means the loss of normal allele of the heterozygous cell occurs that can lead to complete inactivation of the tumor suppressor gene and contributing to cancer progression. Especially in case of clinical relevance, this LOH is strongly associated with this tumor suppressor gene, and RB1 is the here LOH leads to retinoblastoma, and TP53 LOH contribute to multiple cancers. Then BRCA1 contribute to increase in the breast or ovarian cancer risk. Then coming to this clinical relevance or the pharmacological relevance, this loss of heterozygosity is important in cancer diagnosis and prognosis, biomarker development, targeted therapy decisions. Majorly, for example, tumors with LOH in a DNA repair gene increased sensitivity to synthetically lethality concept will be there. That is PARP inhibitors and detection methods mainly by SNP arrays technique, next generation sequencing that is NGS, then microsatellite analysis. So you have to remember regarding loss of heterozygosity means loss of one allele in a heterozygous state, and that is critical for the tumor suppressor gene inactivation. It can may represent the second hit in cancer development. Multiple underlying mechanisms are there. So the loss of heterozygosity is the loss of the normal allele in a heterozygous cell that can lead to complete inactivation of the tumor suppressor genes and contributing to the cancer progression. So this is the picture which representing the maternal and paternal heterozygous copies are forming, and also representing the loss of heterozygosity. That means here there will be a missing of or the loss of normal allele in the heterozygous cell which occurs. These are including the translocation, that is the unusual rearrangement of the chromosome. It is basically detected by cytogenetics or the karyotyping of the affected cells, the type of chromosomal abnormalities in which the chromosomes get breaks and portion of it is reattaches to a different chromosome. Here is about the translocation. Genetic alterations in the DNA can lead to cancer when it is present in a proto-oncogenes or the tumor suppressor genes or the DNA repair genes. The chromosomal translocations in certain cases that can result in either in the fusion of the genes or in bringing genes close to enhancer or the promoter elements, hence that are leading to the either altered gene expression. Picture representing the translocation, the chromosome before the translocation, and there will be a reciprocal translocation taking place. Chromosomes will be, this is how look the chromosomes after the translocation. This is about trinucleotide repeat disorder. They are a set of genetic disorders that are caused by trinucleotide repeat expansion. It's a kind of mutation in which it repeats of three nucleotide, that is trinucleotide repeats increase in copy numbers until they cross a threshold above which they become unstable. So the trinucleotide repeat disorders are the group of genetic conditions. They are caused by abnormal expansion of a specific three nucleotide sequence, that is a triplet repeats within a gene, leading to a gene dysfunction or a toxic protein get affect. So here the DNA contains repetitive sequence such as CAG, CGG, CTG, GAA. So in case of normal individuals, it is repeats are stable and within a threshold. But in case of some disease conditions, the repeat will expand beyond a critical limit. So this expansion is called dynamic mutation, And this major mechanism of expansion which occurs due to DNA polymerase slippage during the replication or defective DNA repair mechanism. So that can lead to progressive increase in the repeat number across the generation. So this explains anticipation, that is earlier onset and increased severity in successive generations. And there are different types based on the location. In that, coding region expansion will be the, so typically CAG repeats produce elongated polyglutamine proteins that can lead to toxic gain of function. Example, Huntington disease are occurs due to this coding region expansion. Then non-coding region expansion is the, that is occurs in promoters introns that can mainly caused by gene silencing and RNA toxicity. That is example for this myotonic dystrophy. Here CTG gets repeated, or fragile X syndrome. In that, CGG repeats in fMRI gene. And mainly thus molecular consequences are include abnormal protein aggregation, and transcriptional silencing will be the example. Methylation in fragile X, RNA mediated toxicity can occur. So generally we can say there are clinical features like neurodegeneration that occur due to nucleotide repeat disorder, muscular dysfunction, cognitive impairment, progressive worsening are also occurs. So here, about its clinical or pharmacological significance, there is no definitive cure will be the mainly symptomatic management only the for the trinucleotide repeat disorders. So this is emerging with gene silencing, antisense oligonucleotides, then RNA targeted therapies are emerging, and CRISPR based genome editing. Gene editing is also coming up with the emerging strategies. And mainly this mutation type is a repeated expansion. The major nature is will be unstable, and it can anticipation, it can gain or loss of function can occur. And also major organs will be affected, the CNS and muscles. So trinucleotide repeat disorders are basically caused by expansion of the triplet consequences, sorry, uh, triplet sequences in the DNA, and that can leading to progressive diseases due to protein toxicity or gene silencing, often showing anticipation. So the trinucleotide repeat disorders are occur when a short DNA sequence, that is a three nucleotide, example CAG, CGG, CTG, GAA is repeated excessively. Normal repeats are stable, but once they cross the threshold, they become unstable and pathogenic. Repeated expansion during DNA replication due to the slippage or the faulty repair. And larger expansions are earlier onset, and moreover severe diseases can occur. So about this, anticipation means disease severity increases and age of onset decreases in successive generations. And different types are there. Coding region expansions are there, that is polyglutamate diseases. In that example is the Huntington disease, that is CAG. Here the CAG will be repeats, and abnormal Huntington protein will be gets repeated. And clinical features are chorea, cognitive decline, or psychiatric symptoms can occur. And next example for the non-coding region expansion example is fragile X syndrome. That is here CGG gets repeats in fMRI gene, and Friedreich ataxia. In that case, GAA repeats in intron of FXN gene, and progressive ataxia can occur cardiomyopathy and diabetes. And these are the common example for the diseases. Then next is the myotonic dystrophy type 1. Here repeats the CTG in DMPK gene, and muscle weakness can occur. Myotonia, cataracts, issues are caused. Then spinocerebellar ataxia. Here, various often CAG get repeats, and it can affect the multiple gene. Progressive cerebellar dysfunction can occur. Wide clinical variability can occur. So that's all regarding the topic of altered gene function. We have discussed different types of mutations, and in that each one explained very well, and loss of heterozygosity. Then in that then also we have explained about the gene amplification, then trinucleotide repeated disorder. So that's all. Thank you.